Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thank you all for coming. My name's Simon Mercer, and you're attending the Brown Bag session on the Microsoft Biology Foundation. So I'll be talking for about the next 40 minutes or so. I don't think I'm going to fill the full hour slot. What I'm going to show you here is I'm going to describe a little bit about the background, why we did a Biology Foundation in the first place, and the different motivations for that. Then I'm going to go on to give you some information about the Microsoft Biology Foundation version 1. And then I'll finish up by showing some demos of what we've actually built. So let me just launch into the first part of that. Essentially what we were trying to do is we were trying to create an open source framework of bioinformatics functionality that could be freely available to everyone who needed such a framework. Now the purpose behind this, just to break it down, we wanted it to be open source because we want other people to collaborate with us, we want to build it in collaboration with others, and we want other people to be able to use it, whether they're in the commercial world or the academic world, on an equal footing. We wanted to do a library because, well, we thought about the community that we were trying to address here. And essentially the way it works is the biological community is very fragmented. There are lots of small groups with very specific needs. If we actually created a set of applications to serve those needs, we would be on the hook for the maintenance of hundreds or possibly thousands of different applications, which was a huge workload for anyone. On the other hand, if you give biologists a copy of Visual Studio, sit them down and tell them to go read the manual, then you're putting in front of them an extremely high barrier to actually creating computing applications that are useful to them. Computer science is a skill in its own right, and if you're a biologist, you're not a computer scientist necessarily, although you probably know most of what a biologist wants to see because that's what you practice every day, but you don't necessarily know the best ways to implement it. So if we're not going to give them if we're not going to give them Visual Studio, and we're not going to give them a set of finished tools, is there something that we can provide which perhaps lowers the barrier to building your own tools? And that's the point of this. This is a library of bioinformatics algorithms and other functions which are exactly that. We've pre-written chunks of functionality for you so you can build the applications more quickly. Perhaps it's useful to go back and think a little bit about the motivations for why we would do this. And the first thing I would do is just showing this rather complex diagram of the genomics and proteomics domains. There are different types of activities occurring under the broad heading of bioinformatics, and they go all the way from examining and assembling the DNA sequence that comes straight out of a DNA sequencing machine through to inferring the function of genes, determining their expression levels, the location of the gene products within the cells, and their function and their effect on an organism as a whole. We're starting at the very beginning of that pipeline, and MBF, the Microsoft Biology Foundation, is represented, the placement of it is represented by the double-headed arrow there on the left of the screen. Just above it, you will see another arrow, ALS. That's the Amalga Life Science product from Microsoft. Microsoft has a healthcare business, a healthcare solutions group, and their primary offering at the moment is Amalga, the hospital information management system. So if you're dealing with patient records, large-scale clinical data, then there's a hospital information management system you can buy if you're a hospital, which will automate that process, the process of collecting the information and of presenting it to you, the clinician. That's a very big product. It costs a lot of money. It's intended for the enterprise. And Amalga Life Science moves one step further down from that, and that deals with discovery informatics data from pharmaceutical companies. It adds a semantic layer on top of gene expression information, gene information, patient information, and things like that. It allows people to explore that information, to aggregate it, and to reason over it, to extract new knowledge from the information related to drug discovery. Now, what we wanted to do is we considered the space that lay immediately before that, even closer to the instrumentation which generates the raw data. We're interested in anything that comes out of a DNA sequencing machine through to contiguous, fully annotated DNA sequence. the very earliest stages of the genomics discovery pipeline. Now, that's an area where open source is very common. A lot of algorithms are developed in that area by academics who prototype them or generate full-fledged applications. They make them available perhaps under licenses that prevent commercial use. Sometimes they're available for commercial use and there's a different license for commerce versus non-commercial use. Sometimes it's GPL and that can cause companies like us some problems if we deal with that. Sometimes they're released on a website somewhere and there's 7 users worldwide. It's an environment where there's a lot of different standards. Everybody would like everyone else to adopt the standard that any given academic has created. There's a tremendous diversity in the different applications that are available. They're licensed in a tremendously broad range of ways, and they're on the cutting edge. Now, pharmaceutical companies, when they're doing their discovery informatics, can't ignore these things because very often they represent the latest developments in their field. However, there then becomes a problem because they might have a large enterprise-level system which is supposed to manage all of the information of their discoveries. They need to integrate it with this rapidly changing open-source ecosystem of academic code. Now, we saw an opportunity there for NBF. That can live in that space. It can provide an easy on-ramp into the larger systems, which include Microsoft systems, but can be others too. But it can also consume data from this very broad and rapidly changing world of file formats and idiosyncratic academic software that represents the cutting edge of research. So what we represent here with the NBF arrow is we're in the genomic space. We've already done some of this, and I'll be showing you our progress in a minute. But we're also a little bit in the functional genomic space, dealing with GWAS, as is listed over there, which is genome-wide association studies, determining the relationship of genomic polymorphisms to susceptibility to disease, sensitivity to drugs, etc. So extracting knowledge from the human genome. But primarily, we're rooted in this most basic form of digital data that comes off sequencing machines, A's, T's, C's, and G's, DNA sequence. Well, we spoke to our customers, and in this particular case, our customers are academic researchers. This is a Microsoft research project. It's based in external research. External research is the same group that are organizing this meeting, and so you understand that the job of external research is to show academics what Microsoft can offer in the research space. This happens to be showing biologists what Microsoft can offer. So we went to the labs and we looked at what is already in use. What we found is a lot of Perl and a lot of Python, a little bit of Visual Basic, a set of tools which are not strongly typed, are created by postdocs typically. The postdoc leaves, the tools stop working one day, nobody knows how to maintain them. So there are a lot of problems there. They create script-based pipelines. There's a lot of error in those. DNA sequencing is changing very rapidly. The first generation of DNA sequencing using a technique called Sanger sequencing tended to produce reads on the DNA molecule, strings of A's, T's, C's, and G's of about 600 to 1,000 characters in length. Now there's a range of second-generation sequencing technologies which are generating in 2 days and for $50,000, or maybe even less than that, the same amount of information that the Human Genome Project generated in 10 years with 23 labs at the cost of $500 million. So the world has changed in only 10 years, and it's about to change again. There's a new generation of sequencing technologies which are, in terms of their data flow, are dwarfing the existing ones. And then you come back to the fact that this data is being handled with Perl scripting pipelines. The two worlds are clearly going to collide at some point, and scientists are very well aware that they're going to have problems dealing with this large amount of scientific data. And perhaps we can provide some help there. So we recognize there are pain points in the academic community around the quantity of scientific data being generated. The tribal knowledge that I refer to on this slide refers to this postdoc problem as I describe it. You get a postdoc in, they turn out not to be very good at the wet bench, they see the computer in the corner of the lab, and they start hacking out solutions on that computer. They're trained in biology, and they're not trained in computer science. The result is code that, even though it might extremely well mirror the needs of the scientist, is poorly architected, poorly documented, and very often poorly implemented as well. And so when that postdoc leaves, it's as if that code had never been written. Somebody else has to find a way of fixing that problem. Now, if you're NIH, you would love to find a way, or NSF for that matter, you'd love to find a way that your funding dollars didn't go into re-implementing the wheel every time a new grant came up. Wouldn't it be nice if you actually could rely on a set of basic functionality that would write the first 20 to 30% of every application, and you could simply just download it, rely on it because it's well architected and well documented so you can understand how it works? Well, we'll be seeing how we got on with that in a minute. Our target scenarios when we were designing this and building this were we were trying to make it easier for scientists to get what they need more rapidly. These early stages of the DNA sequencing pipeline have been performed for 2 decades now. So really, it's not rocket science anymore. It's not really in the domain of scientific research anymore. It's become commoditized, and you should be able to rely on certain standard steps that will extract the majority of the correct information from the data. The data no longer costs that much because $50,000 for a whole human genome is relatively cheap. I believe now you can actually find claims that a whole human genome can be done for under $10,000, and the $1,000 genome is expected to happen in 2 to 3 years. So with prices collapsing at that rate, you can get 3 billion base pairs of human DNA for $1,000. All of a sudden, if you find some problem there, you need to reanalyze the data. You don't need to keep all huge hard disks somewhere with all of the basic data on it. You just take another drop of blood from the same patient, sequence it again. It's just not going to be that expensive. So, rapid application development is valuable for scientists because it means that more time is spent actually using the applications that can extract new knowledge from the data rather than building the applications. We also want to ensure that we can do things like cross-species comparisons. We went down to the University of Washington, they're just local here in Seattle, and we talked to a group of genomics experts there, led by Evan Eichler in his lab. They happen to be working on exactly this scenario of comparing different different genomes from different species, looking at their similarities and their differences. And so we were very interested in learning about that so that we could provide the tools that would enable them to do their science more rapidly and build them into the Microsoft Biology Foundation. So comparative genomics is an area that interests us very much in terms of it's just been one where we've been able to get a lot of information. We're also interested in single nucleotide polymorphism identification. This is a change of an A, T, C, or G in the genome. Single nucleotide polymorphisms are one of the major contributors to why we don't all look the same and respond the same to drugs. They're responsible for a lot of the variation between individuals. If you understand patterns of single nucleotide polymorphisms, which ones you inherit from your mother or your father, then you might be able to understand, or you can gain some insights certainly, into sensitivity to drugs, inherited diseases, etc. And so being able to identify those polymorphisms in a strand of DNA becomes very valuable. And then being able to identify which sets of polymorphisms, called haplotypes, travel around together and are inherited separately. So we've been working on haplotyping as well. So what do we offer people? Well, I think I've spoken about most of this. Essentially, the programmers that are providing a service to the scientists get more of their stuff pre-written. They can build their applications more rapidly. What we've done is we've also been able to demonstrate the value of other Microsoft technologies by integrating them into the Microsoft Biology Foundation. I'll be showing you some examples of that later. If you're a developer, there's no price better than free if you can get all of this just because you can go to a site and download it, and you can use it whether you're commercial or academic, and you can then license any subsequent application that you generate using these libraries under any terms. Well, that's very nice. It's also adaptable because if you can then contribute your code back to that library, then other people can benefit not only from the initial library but from the advances that you've managed to make to that library. For the biologist, they see their research move more rapidly. They see more results for less time and for less money. And we can also show that there's some benefit to using the desktop in genomics research. A pattern that we found time and again is that biologists will surf the web using Internet Explorer. They will read their email using Outlook. They will present their slides like this using PowerPoint. They'll write their papers using Word. And then when it comes to the real science, they'll go onto their Linux cluster and run their applications. Well, you know, in terms of processing power, the desktop is a tremendous machine these days. You know, mine has 4 cores. It runs at, I don't know, 3 gigahertz thereabouts. It's got gigabytes, if not terabytes, of disk space. So, you know, it's a machine that's easily capable of doing science. And so why not build applications that you can access on the desktop straight away? Doesn't mean that you've got to throw the other applications away, but if you're sitting doing nothing but browse the web on a machine with 4 cores, you're wasting a lot of processing power, and why not use it? As well as that, a lot of the instrumentation that we're talking about, DNA sequencing machines, they come with PCs attached. And then there's usually an effort by the informatics groups to move the data off those PCs onto the Linux clusters for further analysis. Why not do the analysis closer to the machine? So, we also, of course, being Microsoft, we're interested in seeing whether other groups can learn from what we're doing here. And so there is a potential, if we had a Microsoft Biology Foundation, for transferring this into other groups. Now, that doesn't mean that this code is going to become a part of a product, but what it does mean is that we can exercise other pieces of the Microsoft product suite, if you like, by applying MBF to those areas. One of them is the Amalga Life Science product, which I've already talked about. We can provide a smooth on-ramp for people scaling up from small work in single labs all the way up to the big enterprise level that Amalga Life Science deals with. The high-performance computing folks have actually been spending a lot of time talking to open source, groups that have been producing academic applications and releasing them under open source licenses around the bioinformatics and genomics space. And they've been persuading them to include build targets for the HPC cluster among their other offerings. They've been very successful with that. Now, if they then expose those services, those applications through web services, we could link to those and build a set of HPC applications. We're also interested in finding out whether MBF can run effectively on a cluster as well. And then there's Azure, of course. Everybody's talking about the cloud, just like everybody was talking about the grid, just like everybody was talking about clusters, but now it's the cloud. The cloud gives us the possibility with very large numbers, huge numbers of commodity machines that you can scale up workloads so long as you have a highly parallel application that you want to use. And so we're looking at ways of building services in the cloud that can be accessed from the desktop, and using those and producing these as prototypes so that other people can go there, either take their credit card, create an Azure account, load the service into that account and run their own service, or they can take a look at the code and understand how to build Azure applications, or Azure services, I should say. So that's why we did what we did, and this is what we did. It's my pleasure to announce that on Friday last week, we released version 1 of the Microsoft Biology Foundation. It's been a long road, and I'd very much like to thank the other people in my group for doing this, especially Mike Ziskowski and Bob Davidson, but there are many others that were involved. Version 1, because this is an open source project, version 1 is effectively just a snapshot of the source tree. It's a well-documented and heavily tested snapshot of the source tree. It's a snapshot that was taken once we'd got all of the features that we decided needed to be in version 1 actually implemented. But work is continuing. We've started on version 2 already in many ways. And you can go and you can take a look at our work because it's all on Codeplex, and I'll be talking about how you can do that in a minute. So what did we actually do? Well, we did exactly what we said we were going to do. We built a set of .NET implemented functionality relevant to bioinformaticians. It's all on .NET 4, so we can leverage the latest features of .NET. It's released under the MSPL open source license, similar to a BSD license. It's available for commercial and academic use, free of charge. I should add a little legal disclaimer to say, please read the license if I'm not giving you a particularly accurate view. It's only 6 lines long or something. It's not a problem. It's an OSI-approved license, so we're truly open source. It's a foundation on which other tools can be built, so it does indeed do that, abstracts out the bottom 20% of everything you need to do if you want to implement new bioinformatics applications. And it provides a set of components, and what it provides is a set of file parsers and formatters, so you can read files, you can round trip between one file format and another. It supports DNA, RNA, and protein sequences, their assembly and their manipulation and their alignment to discover evolutionary similarities and differences between species. I have an example of that I'll show you in a minute. It provides a framework for algorithms. Now, primarily what we're doing at the moment is we're implementing standard genomics algorithms around alignment and assembly of DNA sequences. The reason why standard algorithms is there's no secret source here. What we've done is we've done the implementation, and we've included the reference, the literature reference, to the paper that describes that algorithm actually in the code. So you can see it, and we've made sure that it's consummately well documented so that if you want to use this as a teaching tool to show other people how to construct applications using C# and .NET, it's there for you, and you've got a link straight to the literature so you know exactly how it's supposed to work. We've also provided a web connector framework. Microsoft doesn't want to become NCBI, the National Center for Biotechnology Information. NCBI is doing an excellent job storing large databases of all the DNA that gets sequenced in the world. We're not going to curate that and maintain it as effectively as they are. It's their job. They're the community-accepted standard, along with the DNA Databank of Japan and the European Bioinformatics Institute. Those three institutions around the world are the canonical repository for DNA sequence information, and they expose a service called BLAST, the Basic Local Alignment Search Tool. And they run that very efficiently on a cluster. They expose it as a web service, and we consume that web service. So we can actually access that directly from the desktop without going to their website. And we've done that as part of an extendable framework of web service connectors, so that if we're not connecting to your favorite web service, that's no problem. All you've got to do is look how we implemented BLAST, and you can add your own web service alongside it. Or if we don't parse a file format that's your favorite file format, again, that's no problem. We've made it very easy to build more parsers as a plug-in model and add them to the basic set that we support in MBF. Now, because this is community curated, and I'm kind of jumping forward a couple of slides, I'll probably get to it in more detail in a minute, but this is community curated. What that means is that if you want to develop new pieces and contribute them back to the MBF framework, we'll incorporate them into that framework, and we will release it again as open source. So over time, the snowball rolls downhill and gathers momentum and gathers size, and MBF might start as a relatively small set of functionality, but it can be extended by the community in the directions the community most cares about to benefit scientists the most. So what is it intended to do? Well, I talked about this, so I'm not really going to spend much time on it. It's primarily focused on genomics. In other words, it deals with everything that comes out of a DNA sequencing machine through to contiguous fully annotated sequence. We don't cover all of that space yet, but we've just finished version 1 and we're moving on to version 2, and we'll be filling in some of the gaps as we go. It's important to say at this point that we try not to implement features that we think are good ideas. What we try and do is we implement features that the scientists tell us are good ideas. If we don't have a good use case founded on real scientists who are going to use it, we don't implement it, whatever it might be. We have a finite amount of time. We want to make sure that the features that we use are actually getting, the features that we build are actually getting used. There are other biology frameworks out there. There's BioJava, BioPerl, BioPython, BioRuby, etc. We were going to call this bio.net, but for various legal reasons that name has been taken by others, so we can't do that. So the Microsoft Biology Foundation it is. We believe that we offer a reasonable alternative to these other frameworks. These other frameworks tend to have suffered in some ways because they've been around for 10 years. 10 or 20 years in some cases. On the one hand, that means they have a hugely greater range of functionality than we have if you download them today. On the other hand, they've grown organically for 10 or 20 years. And I've spoken to many of the maintainers of these libraries, and in some cases, they really wish they had the opportunity to start from scratch. We're coming into the genomic space much later. Version 2 of DNA sequencing is now available. Third generation is coming soon. Many of the initial problems have been solved. Many of the very broad range of file formats that are supported by these other libraries have subsequently fallen out of favor with the community. People tend to have settled on FASTQ, SAM, BAM, and a few other formats. And we support that relatively narrow range of formats, although, as I say, it's extensible. So we've learned to a certain extent from their example as they've forged ahead in the bioinformatics field ahead of us. There are also some unique or some unique benefits from using Microsoft platform here. We have a very powerful set of visualization tools, and over this summer we have some interns who are experimenting with using Pivot and Sea-Dragon and Deep Zoom to visualize genomic data. We hope to see the results of that soon, guys. Yes. We also have things like .NET Parallel Extensions, which is a .NET 4 development. What that does is it automatically senses the number of processors and cores on the machine on which MBF is being run, and it will parallelize out an algorithm to take advantage of the full power of that machine. That means that what we've done is some of the algorithms that we've implemented are not the absolute standards that I described earlier. They're parallelized versions, so they take advantage of the machine on which they're run. Yes, large data sets. We try and deal with large data sets effectively. We largely succeed. It's worth mentioning a few things about the design goals of what we did here. What we've tried to do is we've tried to make it extensible. As I say, we don't implement everything, and so what we do is we provided a plug-in architecture so that you have a set of APIs that you can code against, which make it very easy to extend this in new directions. This is good because then we'll accept the contributions if you do extend it in new directions, and we'll bake it into the next version of MBF and release it as open source. It's language neutral. Because it's built on top of MBF, it supports all languages the Common Language Runtime supports, which at last count was around about 70. So if your programming style is Visual Basic, that's fine. You can contribute in Visual Basic. If you prefer Python or a functional language like F#, then those are also available to you. So you can develop on this in a way which results in all the languages being equally compatible, and you can develop applications or make contributions to the core libraries in whatever language most suits your programming style and the nature of the problem you have to solve. Because we're built on top of .NET, we can also be assured that we're in the mainstream of Microsoft technologies. What that means is, wherever Azure goes next, you can be pretty sure that .NET assemblies will just work out of the box. If you're interested in combining MBF with SQL Server, SQL Server has its own CLR within it, and you can add some code. You can decorate some of the classes in MBF, and it'll actually run inside, or some of the classes will run inside the CLR, so you can actually integrate MBF with your database. And there's a range of other things as well that you can do in that space. Interoperability. Well, it's not really true, this last statement on the slide. If I'd noticed it when I was looking through the slides earlier, I probably would have edited it out. But it does give me a segue into what we're trying to do here. We're very much aware that science isn't a Microsoft monoculture, that there's a very wide range of other platforms being used, principally Linux, OS X, etc., in science. If we're going to be useful, and that's our goal here, is to be useful to scientists, we have to be interoperable. And so what we're doing is we're evaluating MBF to see how it would work with the Mono project, open source implementation of .NET, to see whether we can be cross-platform. Also, anything that exposes itself through web services, we can consume because of our web service framework. And so what we're trying to do is we're trying to deliberately build this in a way which can access other tools on other platforms. In terms of non-Microsoft tools on the Microsoft platform, that's also possible because, as I say, we have a plug-in model. And so when we extend, we don't currently have supported databases, but you can bet that when we build that part of MBF, we'll support SQL Server and we'll do it very well. But if you have an Oracle database, well, it's a plug-in model. So if you want to write a plug-in that accesses your MySQL database or your Oracle database, I would invite you to do it and contribute that plug-in back to the project, and we will release it as open source alongside the rest of MBF, because we do accept that people should use Microsoft because it's useful, not because you feel locked in. So what do we actually have? Well, it's pretty straightforward. This little diagram, it's not a comprehensive architecture, but it gives you some flavor of what we're talking about here. It's a set of four things. One is an internal object model that models the biological objects we've been talking about here, like proteins and peptides and DNA sequences and RNA sequences, and the different encodings for those things. We have a set of file parsers and writers. we have a set of algorithms, and we have a set of web service connectors. That's all that NBF is. And it's important to point this out because I could stand up here and page through code all day and show you a library, but it's not very exciting to look at. So we built some applications that demonstrate that functionality, and that's what I'll be showing you in a minute. But NBF itself is not an application. It's a foundation, and you can build your applications more rapidly with it. Here's an example. Well, I'm going to show you a demo anyway, so I'll skip that one. And I'll skip that one too. So how do you get NBF? Well, we launched this web page, research.microsoft.com/bio. That is a link into the sort of work that we're doing in this space. Now, you will notice that it says Microsoft Biology Foundation at the top of this page. If you go to the /bio link, it'll take you to the Microsoft Biology Initiative page, which is slightly different. The Microsoft Biology Initiative is a slightly broader effort than just the foundation. And the way it breaks down basically is the Microsoft Biology Initiative consists of two sides. One side is the Microsoft Biology Foundation. That's the set of code I'm talking about today, the basics implemented well, we hope, that are available as chunks of functionality that you can incorporate into your application. But on the other side, we have Microsoft Biology Tools. There are a set of tools that Microsoft Research has produced over the years that are directly relevant to biological research. Some of these are actually built on top of the Microsoft Biology Foundation, and in time we expect a greater and greater number of those tools will actually be built using the Microsoft Biology Foundation because NBF has been adopted and is being used by our researchers actually in Microsoft Research now. So we have these two sides. You've got the .NET assemblies of the foundation, and you've got the full-fledged applications of the tools side, and collectively they form the Microsoft Biology Initiative. So you can download it all from our website just there. It's licensed under MSPL. It's very easy to install. We've taken some time to create installers. They detect dependencies. If you don't have .NET 4, they'll point you to the right location to load those. We have other tools available there. And I'll be talking about those in a minute. The documentation is all there. I can kind of skip that. The source code, however, doesn't live on research.microsoft.com. It lives in Codeplex. Codeplex, as you're probably aware, is a source forge that's run by Microsoft. And we found that it hasn't been the world's easiest source forge to use, to be perfectly honest. But what we are doing is we do our development using Team Foundation Server. We do it in the open. On Codeplex, so you can go to this site, you can browse the code through this site, you can download the source as a zip file, you can participate in the conversations we have with other researchers and developers around the world who are helping us develop Microsoft Biology Foundation or who are contributing code to us. And you can also apply through that site to become contributors yourselves if you're interested in adding anything to it. So that's all on Codeplex. As I say, we've adopted an open source strategy, but we've actually been blazing trails inside Microsoft in this space. We're not the first open source project from Microsoft, far from it. But we are an unusual project in that we're community curated. We encourage contributions from the community, and the community is, of course, not necessarily entirely created of Microsoft employees. Which means our legal department has fits from time to time because we're bringing in code from the outside that Microsoft does not own. That's okay because it's going out as open source, but nonetheless, it's an interesting little journey we've been on. Codeplex doesn't really support this very well. They support single branch development, and we'd like to branch. They don't support the full range of TFS, and we'd like to use far more features in the Team Foundation server than are exposed through Codeplex. One of the biggest problems we have is when third parties do contribute code to us, we want to be able to hold it and review it and check it for quality and make sure it complies with the standards of the rest of NBF prior to integrating it. And that's not really very well supported on Codeplex. So we're looking for other source forges, and we're also considering, alongside a move in terms of the source forge that we use, we're also thinking of perhaps a new home for the Microsoft Biology Foundation. And so I might be back here next year just talking about the Biology Foundation, because if it's not located in Microsoft for various legal reasons, we can't actually label it a Microsoft thing. If we do move to another foundation, it'll remain open source, and we'll make sure that foundation still supports all of the ideals of the project that we've created. This should be free for scientists and commercial users alike. We're looking at the Apache Foundation, among others. Essentially, it'll provide us with a legal umbrella that separates this a little bit from Microsoft, because as we get more contributions from the community, although Microsoft will stay engaged, we're no longer the only game in town. Our licensing has also proved a little interesting. Microsoft is, of course, a company that sells products. And if we're trying to do open source as well, then really, if open source is to be useful in the context of the commercial world, we actually have to be able to release some things and control it, and other things far more openly, and do so under different licenses. And this is what we found. Again, it's caused us to blaze some new trails legally as well inside Microsoft, or certainly in terms of policy inside Microsoft. The Microsoft Biology Foundation, MSPL, completely open source. The sequence assembler, which I'll be showing you, which is one of the demo applications, is also MSPL. Windows Presentation Foundation, .NET, those sort of things. We built it ourselves. But when we actually show you an Excel ribbon, which integrates a lot of this bioinformatics functionality directly into Excel, Microsoft has patents and intellectual property that it doesn't want to open up to the rest of the world by making that open source. So what we had to do is use another license, which does allow you to get the source code, but it's not OSI approved open source. And that's the MS-LPL license. What that does is it allows you to look at this stuff, implement it any way you like, but it has to be on the Windows platform when you've finished implementing it. So we've had to kind of trade off some of the pure open source ideals for the realities of working in a for-profit company. The show runtime is an advanced math library that we've also made available. It's available for download. You can read about it online. It was made available to us so close to release that it's not fully documented, but it actually provides a wrapper for the Intel math kernel libraries. We will be using the advanced math functionality that makes available in a subsequent generation of demo applications that are based around David Heckerman's work on HIV and vaccine design. And we've released that under a custom license because, oddly enough, Intel doesn't want us to make all of their stuff open source on their behalf. I'm just going to move on and talk about the community. As I say, we no longer think of this as a Microsoft-only effort. It was conceived to move outside of Microsoft and get a life of its own, although we will stay engaged with that project as it moves on. In consequence, we've set up a technology advisory board. There's 9 people on this list, and they come from, 3 of them are from the academic world, 3 of them are from the commercial world, and all of the organizations they represent have now adopted and are building actively on MBF and are contributing code back to us and are using MBF in applications both in-house and in products in some cases. And as you can see, Johnson & Johnson Pharmaceutical Research and Development is one such group. Illumina Corporation, the foremost manufacturer of DNA sequencing machines, is another one. University of Washington, and internally within our own Microsoft Research eScience group, David Heckerman's team et al. We're trying to find ways of being faster in terms of taking in external contributions. Frankly, that took a backseat while we were pushing to get version 1 out, but now version 1 is in place. We're starting to look again at better ways of bringing in stuff, quality controlling it, and getting integrated quickly. Because we're also finding that when I give talks like this, and there are Microsofties in the audience, you'd be surprised in a company, well, you probably wouldn't be surprised. It's a company of 90,000 people. There are people out there who were biologists in previous lives, and they're only too delighted that they get an opportunity to fall back and work on their hobbies in their spare time and contribute to a Microsoft project along the way. So we actually have a range of people. Somebody actually implemented an entire suite of phylogenetics algorithms over a long weekend, and we haven't got that incorporated yet, but we've actually sparked a lot of interest inside Microsoft that has nothing to do with product, just a bunch of geeks playing with biology. So, the bottom line, we're open source, we're community developed. Please come, take a look at the website, contribute code if you find it interesting. If you don't find it interesting, it doesn't matter, download the code and use it for whatever you like, if it's useful to you. We're going to do periodic releases, so there will be a version 2. But over time, what we're going to be doing is we're going to be transitioning progressively more into the community. The technical advisory board that I just showed you owns the version 1 release in effect, and they'll become more involved in promoting it in the community and being visible ownership as we move outside of Microsoft with this project. Now, for as long as there's an external research team, and for as long as that external research team has a healthcare interest, we'll be doing projects in software that involve biology. So we'll be using MBF and we'll become active contributors to it alongside everyone else. That doesn't necessarily mean that we'll steer its direction in the future, because the commercial world and the academic world will both implement their own features and request features from us, and we'll be implementing them alongside our commercial and academic partners. So I'm going to show you a couple of demos. I've told you a lot about what it is. Now I'm going to show you it in reality. Okay. So one of the things that we've done is we've created a DNA sequence assembly tool, and I'll just show you what that really means. I'll choose some short-read DNA sequence information, and I'll launch the assembler. This is a Windows Presentation Foundation application. It was created by us, and it essentially only exists for one reason, and that's to show off what MBF functionality is. So don't forget, we're talking about library here. We've exposed a selection of the functionality in here to show you how you could build your own applications. Oh, this is open source too, so if this looks useful to you, you can download it, hack it around, and your users can use it too. What you're seeing here is a set of short reads, which are small fragments of an original DNA molecule. The DNA sequencing process takes the original molecule, shatters it randomly into pieces, and then those fragments get reassembled. And of course, then what you have is the problem of taking all of these fragments, overlapping them appropriately, and reconstructing the original DNA molecule. Now, different sequencing technologies will produce you fragments of different lengths. Sometimes there are some additional wrinkles and interesting bits of information, like you have a piece of DNA this long, and you've read a bit of information from that end and a bit of information from that end, but you don't know what's in the middle. So there's all sorts of variations. It's actually quite an interesting field. Here's some short read information. These reads tend to be about 35 base pairs long, which are the A's, T's, C's, and G's that make up the alphabet of DNA. So what I can do is I can assemble them together. And I'm going to choose an algorithm from this drop-down. These are different ones that we've implemented. I'm going to choose the Smith-Waterman algorithm, which is a standard algorithm. It's been around for 25 or 30 years now, that will assemble those fragments, find those overlaps, and reconstruct the original DNA sequence. That was a lot faster than I thought. It's normally about 15 seconds. Never mind. Okay, well, I'm pleased it worked. So this is a trivial amount of data, because I didn't think you'd appreciate sitting there for 10 minutes to watch me do some more serious piece of data. But as you can see, we've created a contig, or a contiguous piece of DNA. Now, what you're seeing here in the upper window, these green lines that are under this gray area here, so these green lines here, these are the original fragments as they are aligned. They collectively contribute to this blue line at the top, which is the consensus, or the reconstructed DNA molecule. Now, if you look in this lower window, which, as you can see, scrolls as I move the upper one, you can see the actual A's, T's, C's, and G's from these different fragments, and you can quite clearly see they're aligned. The reason why they're colored this way is so that by eye you can actually spot errors or polymorphisms in these DNA fragments. So as you can see, that goes together pretty quickly. I can look at the report and I can see that took a grand total of 0.44 of a second, which was pretty fast. Now, if it hadn't taken such a short time, I could have pulled up the, what do you call that thing, the task manager, and I could have actually seen that it parallelized out the execution across all the cores in this particular machine. It's a 2-core laptop. It's kind of old, actually. So that gives you a brief idea of what that as a sequence assembler will do, but I can show you another example here, which might be a little more complex. What we have here is a set of fragments of a prion protein from human. The prion protein it's a naturally occurring protein that's expressed in human brain tissue. But what happens for some mysterious reason, which I don't think anyone really understands at the moment, is it can fold in 2 different ways. There's a normal form, a normal conformation, which is normal and leaves you normal, which is great. And then there's a second conformation which results in clustering clumps actually in the brain cells, causes death of the brain cells, and you can get Creutzfeldt-Jakob disease, which is a brain disease which will definitely kill you. There's also mad cow disease, the bovine spongiform encephalopathy, which is another version of this exact same thing. What seems to be the case is that when you're exposed to a protein molecule that's been folded in this incorrect manner, it catalyzes the other protein molecules in your brain to fold in that incorrect manner. And so although this isn't an organism, this protein is an infective agent. This makes prion diseases particularly dangerous because you can actually take infected meat and reduce it to ash, and the ash is still infective. So it's an interesting biological conundrum, if you like. So again, I can assemble it using Smith-Waterman if I want to. And I'll do so just because, although this is longer reads, these are 700 bases long, so it is going to take a bit longer. It'll probably take about 20 seconds this time for sure. These fragments are now being assembled. It's determining the overlaps, and it's orienting them and creating a config. Once it's completed, I'll show you, and you'll see in a moment when it does complete, that these fragments go together perfectly, and it reconstructs the original sequence of this fragment of the prion protein. But there's a trade-off here. This is kind of slow, and certainly if you're standing up here, it seems even more slow than it does if you're sitting down there. But there we are, it's done. And I can scroll through this, and I can take a look at that assembly, and I can look at different regions of it like this. I believe there is actually, no, there isn't a polymorphism that I can easily find, but this data does have a certain amount of error introduced into it to demonstrate that it will actually reassemble data that's in error. But as you can see, everything's gone together. I can look over here, and in this tree I can see contig 1. There's only a single contig here, so all of those fragments assembled together to form a single contiguous piece of DNA. Now what I can do is I can take a look at the report, and I can see that that took 30.5 seconds to execute. So what I could do is I could go back to the DNA sequences here, and I could run an assembly again, a second assembler, and this time I'm going to choose PADINA, which is just available here. Now PADINA stands for Parallel De Novo Assembler, and it is something that we've implemented as part of this. Let me just run it, and then I'll talk about it once it's run. Okay, so I hit that button there. Now what PADINA does, and you'll notice that was nothing like 30.4 seconds, by the way, same data. PADINA is a De Bruijn graph-based sequence assembler. So what it does is it takes each one of those sequences, it breaks it into small pieces. Actually, it moves a window across the sequence. It extracts all of the different, what they're called KMAs, the short sequences that are represented there, and it creates a graph where adjacent nodes are separated by only one base as you move along. Then if you get repeats, you'll get cycles in your graph. If you've got errors, you get bubbles where two paths in the graph diverge and return. You take each one of these sequences, you do that with it, and then there are various error correction things you do to burst those bubbles and break the branches in the graph, and the result is you end up with this overlapping array here. But if you look over in this side, you've got two contigs. One of them only consists of a single sequence, the other one consists of all the others. So one of those sequences didn't go in. Yet we know it's good data. The Smith-Waterman algorithm I just ran showed you it all fits together perfectly. And so what you can see here is that this is another algorithm. You get a huge trade-off in terms of speed, but unfortunately it doesn't, it's a little bit error prone. This points out one thing about all of MBF really, which is that if you need to know what you're doing to do these algorithms. It's very easy to end up with garbage on the screen. It really honestly isn't our fault. That's how the algorithms do work. So you have to understand the data that you're using, you have to understand why you chose that algorithm, you have to understand the parameters that go into that algorithm. So this isn't for the general public, if you like. This is for people who do understand the biology, but we've implemented these algorithms. These are man-months of work, and you can take them for free. And incidentally, this algorithm, the PADINA one, it's very well documented, and you can actually pull it apart and change the sequencing of the different steps that are involved. It's a 6-step algorithm. You can even subclass some of those and roll your own steps, so you can create your own custom De Bruijn graph assembler if that's what you want to do. Now, if I wanted to, I could take the reconstructed DNA molecule here, and I could run it through a web service. And as you can see, I've got a little drop down here to select web services. So what I can do is I can click on this, and I can select one of the basic local alignment search tools. Now these two are two of those 3 centers around the world that I was talking about earlier, the EBI and NCBI, that offer all the DNA in the world that you can go compare that DNA sequence with and see what it was similar to. However, the top one here is different. This is the BioHPC project, and I see Jarek and Bob in the corner there. So from Cornell University, thank you very much. And what Jarek and his team have done at the CBSU is they've ported a range of bioinformatics applications to an HPC cluster, and they make all of that port available to you as open source and all exposed as web services. So what we've done is we've got an example here of connecting to their BLAST web service, but there's another, 26 applications at least? 43. They've been busy. Some population genetics algorithms, some DNA assembly algorithms, and other things that are available free of charge from Cornell, or you can take the code and you can put it on your own HPC cluster, and you can stand up a service that offers similar sort of HPC in bioinformatics from your own site. So I could click on that and run it, but NCBI is up about 70% of the time, and it's slow. So here's one I prepared earlier in the finest tradition of TV chefs. Oh, yes, it has finished. Good. Okay, so what this is, it's a set of windows. You can open them and close them, obviously. So the sequences that were the input are in that pane. The config, the assembly that we produced, is in this pane here. And then down here, we took the reconstructed molecule, and this is the result of the web results. So you can see a set of sequences here. Now, what these are is they're sortable and groupable and pasteable and all sorts of things. But what we've done here is we've gone to the National Center for Biotechnology Information. We've queried this reconstructed DNA sequence against every other fragment of DNA that they have in their database, and their databases are huge. After a couple of minutes, they only return those that match that original sequence within certain specified parameters of match. You then have a set of sequences that are being returned to you that are very similar to the thing you've just sequenced and assembled. So you can then look at the annotations of those things and figure out whether, maybe somebody else figured out the purpose of this molecule that you've just sequenced. Maybe somebody else in the world sequenced the same thing in horses and chimpanzees and fish and fruit flies. And if they did, you could construct an evolutionary tree that shows how this gene has evolved over time. Those sort of things. And you can do that with these sort of BLAST results. You can also visualize them. And for this, I have to thank my esteemed friend here from Queensland University of Technology. And I wish I'd picked a better data set for you because it's not showing this so well. But I can zoom in here. This is a visualization of the data. Now, I'm sorry it's not the most exciting visualization because basically what you're seeing is a big blue ring. This black cloud, because the names are overlapping so closely of the blue dots that you can't really see what they are. But I can zoom in and eventually the dots resolve, but I can't zoom quite far enough to do it. And each one of these dots, what it is you see there's a ring around a central blue dot. What this has done is it's represented all of those BLAST hits spatially. So the dot in the center is the query sequence that we sent off to NCBI. And the dots that surround it, which should really be in more of a cloud, represent all of the other sequences, DNA sequences, that were returned by that query. The distance from that central dot is proportional to the degree of similarity between the query sequence and the other sequences that were returned. And I can find out more about each one of these individual query sequences. I can pop this window open and pin it, and I can click on one of these. Oops, I should probably have the hit detail, that one up, right? I can click on these, and nothing appears. I got it? I got it. Right, good. And as you can see, this is from Alces shiracei, which is that well-known species of something. And this is a prion protein sequence from that species. PRNP tells you it's a prion protein. And I can click over here. Yeah, I don't know what that is either. One of these days, probably random chance, I would hit eventually an organism I recognized. But nonetheless, that's a set of those. So I'm being gestured at from the back of the room, so I think I have to end it there. But please come and see me later if you'd like to see more demos. There's plenty of other things I could show you. I can show you multiple sequence alignments in DNA and proteins, which actually highlight these evolutionary relationships. I can show you the same functionality encapsulated in a ribbon at the top of Excel, so you can do exactly everything you've seen me do here, but in an Excel spreadsheet. And biologists really like that because they very often load data into Excel to take a look at it anyway. I can show you the same stuff encapsulated as individual units of functionality and strung together as workflows and available in Trident. If you've seen our workflow engine called Trident, which is a scientific workflow engine because it captures a lot of provenance, so you can actually do reproducible science and build scientific workflows in Trident. So we have all of those things ready to go, but it's a .NET assembly, so I urge you to experiment, join the community, and see where it takes you. Thank you very much.